Okay, so welcome to the third video in this course G03 on read mapping, and we're looking at here about how we can link read mapping and genome assembly together de novo assembly, and how essentially they're very parallel and integrated processes together. Of course, in both areas, we have the analogy from before that it's a lot like cooking, that we have particular read libraries as ingredients, and then we have particular algorithms or appliances that we can use, and then we have models that will be specific to the context that we're talking about, what recipe is best for us in that situation. And that will affect the output that we get at the end. And a bit like cooking, there are certainly things that are maybe not good, but then having perfection is also more tricky to think about. More widely, we have examples like the Arctic workflow for thinking about how read mapping and de novo assembly work together. So if you look at the nanopore pathway up here, we have this is the Arctic network flow, where we have the creation of reference guided de novo assembly and stop based on creating a mutation file known as a VCF file, as well as fast consensus assembly. And then if we have shorter read Illumina data, that's where remapping might be more effective. After QC, we can map the reads with BWA in this pipeline and identify variants in the same way and do similar things. So these different approaches really reflect different options of the same thing. So we also have other options that are more expanded in this diagram I've shown before. At the start, we have different options for QC. This is for long read data, but it's similar. We have options then for self-correction, read error correction, and then for de novo assembly. There's more error correction here, and then assembly refinement over here, and then our alignment approach. So we're going to try the tool Minimap 2, but there are other tools depending on what you want to do as well. And we're kind of progressively moving through aspects of this quite complex picture. For us, we can see this diagram here. What we can follow through there is our FASTQ files over here, and we do quality control. We may do some of these steps or just one. It depends on our library. Certainly, we'll do FASTQ at the start, maybe FastP to screen the quality, and FastQC again at the end to check that quality. Maybe multi-QC as well to summarize all these HTML files that we produce together in a single report. So from QC, which we see as an essential step, we have optional steps, which are listed here. Certainly we want to do some of these steps, but what exactly we want to do depends on the project. It depends on our recipe and our algorithms and all the rest. So we might remove host or vector reads depending on what we're looking at. So we might therefore map our reads to the host or vector reference genome with Minimap 2 or equivalent, get the unmapped reads, which would be you'd expect it to be the vast majority of them, and then convert that BAM to FASTQ. So we're still dealing with FASTQ files at this point, even if we skip this step and go straight to read mapping or assembly. When we looked through assembly before, we had options like de novo assembly with spades. We didn't do improving the assembly, but there's a myriad of tools that can do that. We looked at Quast, how we can quantify the assembly quality using that tool. And then if we did subsequent step, we could look further at things like scaffolding the assembly using existing assembly, using sequence alignment with MOV, progressive sequence alignment with MOV, annotating the assembly with the likes of Proca or equivalent as well. There's quite a few steps there. On the read mapping side, we're going to look at how we can map a reads to a reference. We're going to use Minimap 2 and another tool. And then we'll look at how we can remove duplicate reads with Picard or on alternative as well. And that will lead us to the next course, G04, where we're looking at this mutation detection area. In our last slide, we looked at deformed graph assembly in terms of assembly algorithms, and we looked at how k-mers were the key unit. And we're going to use similar approaches here, dealing with k-mers because they're more efficient. So k-mers, as mentioned before in the previous video, are used to construct a hash table. And if we're not using k-mer, if we're using seeds, like the Burroughs transform type approach, they're kind of similar. It's just the data structure and the way the data is stored on the computer is a little bit different. So this use of k-mers with a k-1 base overlap makes this whole task more easy because we can store k-mers that are identical together as a single item with mapping to multiple locations across a reference genome. So the different locations are stored and are recorded as being associated with that k-mer. As the k gets more big, remember, we get more uniqueness, but then you'll typically get a longer compute time and lower overall coverage. So there's kind of a Goldilocks zone where we have efficient computing, but a relatively unique k-mer. For virus data, you can use quite a high k-mer and it'll work fine. For host data, you may want to actually increase the k-mer iteratively to kind of understand what is optimal. And in practice, we'll use multiple k-mers to make the best assembly out of that consensus across different k-mers. So these k-mers look like this, that we have our sequence read down here. If we make k-mers of length 5, we'll chop them into bases of length 5 here. So you can see the first 5 bases down here, and then we skip one base and then move on to the next k-mer. So skipping just a single base. Some algorithms will skip 2 bases, but for indexing the reference genome. Here we can be exhaustive for viral sequence data at least. During de novo assemblies, we took those k-mers and then merged them gradually as we looked at the different paths through the different k-mers. And then what we'll do in our next practical is visualize these. So this is our reference genome kind of listed here on top. And then we have this graph showing the read coverage here, which is kind of on the low side here. And you see that across this region we have some letters that are covered more and some letters that are covered less. And each of these read pairs contains a forward read in pink and a reverse read in blue. And then we can look at the different base differences across this whole segment. So you can see that the read depth coverage here for normal libraries does vary a bit. And then if we're looking at transcriptomics, the same sort of thing applies, except for mapping RNA reads to a transcriptome assembly, we'll have to think about how we remove non-coding elements that might affect our picture. So mapping RNA reads and DNA reads, again, it's the same principle, but different application and different details in that context. When we're thinking about using mapping and assembly together, what we consider is our starting DNA here on our reference genome. I may have repetitive segments symbolized with the red sequence. If we have short reads, as shown by the red bases here, where they don't span the full length of this sequence, for a very long repeated extraction, that will be difficult to resolve, and short reads just won't resolve it. We need long reads, preferably ones that span this black to black region. So what we'll get if we try to de novo assemble those reads is a resolution of something like this. So it'll be a very short segment because our reads are comparatively short, and we'll get leftover reads. And those leftover reads can be informative because we can find out what was inside here, that they didn't align properly, didn't align uniquely. And depending on the structure of our reference, we may find that we have something that's different. So you can imagine here in our next examples, if we have this structure BAAAC in either a reference or our sample of interest, how that might affect the reads that we get and also the mapping to that. So we might have low resolution of these 3 repeats here, but we know that there's at least one copy of A, and then we can count the number of reads that are mapping across this whole region to count how many potential copies of A are present. So we can imagine if you had 2 copies of A, that might affect the read coverage. If you have 1, 2, 3, 4, 5, 6 copies of A, and if you had a circular reference or a circular sample, again, that's going to affect what you get. And it's the same sense here that we have certain reads that are forwarded because they cross a boundary, but the blue reads here that move from A into A are not going to resolve clearly exactly where they map, because maybe they map at multiple locations. So you have to bear that in mind when you find uneven regions of read coverage that are maybe a half of what we expect, or twice, or 3 times, or 10 times. That can be informative in determining structural variations as well. So in this way, weave mapping and de novo assembly work together when we think about it. So what we can do here when we de novo assemble our sample is that we can actually map the reads to it. And it's kind of listed here. We have this option of read mapping. We can map reads to this de novo assembly, and this Nature Reviews article here outlines that well, that we might have a process where we QC our reads first of all, and then we de novo assemble, we scaffold our assembly, so for high quality assemble, take our unassembled reads to try to finish the assembly properly, and then we can map our reads to not only a reference genome, but also its own reads as well. So we might get reads that don't align. We can de novo assemble them to work out what they are. An important step here is to consider how we can map our own reads, our own samples reads to its own de novo assembly, and look at what variations occur. So those ones may, for viruses, reflect low frequency mutations, or they may reflect heterozygous SNPs at a host or a vector, a longer eukaryotic genome. So we can think about it like this, that we have this step here, that we can map our own reads to the assembly and quantify the diversity that we see there, and explore this whole area of heterozygous and low frequency variations. So if we think about this, we have some sample that's being mapped to our reference, and that might tell us a few different things. We might identify that we've a homozygous difference or high frequency difference with the number of the fraction of reads here. Fraction of reads with the different dates. With the let's call it the derived allele at that site. And the first site, maybe it's 0.99, because there'll always be some errors in there. Might have another site, 0.22, which again is 0.98. It's quite high. We might have a site 33, and here maybe it's 0.41. Might have a site 44, where it's 0.95. Might have a site, well, let's call it 50, where it's 0.23, and some site 66, where again it's 0.97. And maybe another one is site 77, or 0.99 again. And what we can see here is that there are clear differences where this is a clear base change, because we know that we use some erroneous reads as we go through. And we can look at that pattern across all the different sites that are different, in case there's something unusual, in case maybe this is 0.9 and 0.8 and 0.9 and 0.8, there's some sort of technical artifact we need to look at further. And what you might do is draw a histogram of the frequency of those differences going from 0 to 1 here. And typically, if you're dealing with the eukaryote genome, you're going to expect something like this, where you expect very few, bit of a bump at the 0.5 region, and then a lot more probably that reflect homozygous changes. So this actually would spike up here, and that's around 0.5. For virus or other notionally haploid genomes, you wouldn't expect any distribution, expect this peak at homozygous changes. So there you would expect maybe there may be some low frequency variants, but they would tend to be rare. So you'd expect maybe some changes that are here or there, but there shouldn't be any peak at 0.5. And you'd expect the same higher peak here for homozygous changes. So we can look further at these sites. So if we this is mapping to the reference, and here we can think of, okay, we map it to our self. So map to self here. So it's site 1,1. Assume the coordinates are the same. So here we might have looking at frequency of 0.02, for example, because there's some background error. Here we might have a change here. So let's say here we have 0.48. Okay, that's inconsistent with what we have above. So that might be worth exploring. Here we might get another value of 0.39, and that corresponds with what we have above, so that might be real. This one maybe you get like 0.2, so that's okay. This one, let's say we get a value of 0.01. So that's different. And then let's say we got 0.97, 0.03 here, and 0.99 here. Sorry, 0.01 here. So where you expect for these positions where there's a clear snip where the map reads back to itself, there should be no difference beyond the error rate. So what we might then do is say, okay, for sites 22 and where's the difference? Site 50, I need to look at what's going on. So you might think, is this a false positive snip in our reference mapping? And this one, that appears to be real, because if it's 0.48 when you map it to itself, it's definitely a change. So this might be a low frequency or just a variant within heterozygous variant here, where there's a mix of alleles. So let's say there's a mix of A and T alleles, but that hasn't been detected when mapping to the reference. And that can still happen sometimes. So you need to explore what's going on here. It could be something like this, some sort of TA repeat locally, or another indel that is affecting this location. So that's why mapping to your own assembly is actually really good. If you want high resolution of a particular sample, that is advised. If you're dealing with 1,000 viral samples that you have to get through very quickly, then that might be quite laborious to do all that. So that concludes this part on integrating mapping with assembly. So you can see here that there's quite a few steps to think about. What else you can do with the data is very important. So please see the next video with further details on read mapping.